The overall goal of this glass surface functionalization process is to allow for the gentle capture and release of cells of interest.
This method can help answer key questions in the field of tumor angiogenesis, enabling screening of cell biomarkers that indicate drug resistance. The main advantage of this technique is that it is completely customizable for virtually any cell type of interest. As long as there is an antibody specific to the cell type, the surface can be adapted for its capture.
Though this method can provide insight into cancer angiogenesis, it may also be applied to other diseases as purified samples are necessary for the development of more personalized treatment regimens.
We first had the idea for this method when we were trying to develop a reversible release mechanism for capturing cells, using the interaction between desthiobiotin and the avidin family.
After cleaning a glass surface in an oxygen plasma machine for 5 minutes according to the text protocol, prepare 2.5 milliliters of 2% reconstituted 3-aminopropyl triethoxysilane, or APTES, solution by combining 50 microliters of APTES and 2.45 milliliters of ethanol in a conical tube. Pipette the APTES solution onto the glass surfaces. Then cover the surfaces and place them on a platform shaker at room temperature for 50 minutes to evenly distribute the APTES layer. In the meantime, preheat the oven to 55 degrees Celsius for the 8 and 24 well plates, or 90 degrees Celsius for the glass dishes.